Okay, class, how are you? This is lecture 3, and today we're going to cover proteins. So hopefully proteins don't need any introduction. You guys have probably had these in many classes dating back to high school. Proteins as a class, they're the most complicated biological molecules, and we'll see that today in just a number of different ones. And proteins perform most of the catalytic activity in the cell. There are some exceptions, like ribozymes and things like that, but in general, and for this course, you can sort of think about proteins being, the catalytic engine of a cell. As we talked about last time, proteins are composed of amino acids, and we looked at the list of 20 essential amino acids, and proteins are made by being translated on the ribosome from the messenger RNA that's generated from the DNA. Okay, so if we want to sort of just see proteins in a nutshell, they're basically made of amino acids, okay? And so here you have methionine, aspartic acid, leucine, and tyrosine. And if you guys remember, amino acids, they have an amino terminal end and a carboxy terminal end. And proteins and polypeptides are made by a condensation reaction, where these are joined together by peptidyl synthetase in the ribosome, and water is released, and then you get the formation of a peptide bond. And so you basically have these repeated structures of individual amino acids. And the amino acids are basically all the same along their sort of core, but then they each have a different side chain. Okay, so this is methionine, and shown here in green is the methionine side chain. And here you have aspartic acid, and here is the aspartic acid side chain shown in blue. Leucine side chain is shown in green, and tyrosine side chain is shown here in blue. Okay, and so you get, when the messenger RNA is being read on the ribosome, you actually get the formation of this peptide bond between the individual amino acids that make up the polypeptide or the protein. Okay, and so just like with DNA, proteins are directional. You have an amino terminal end and then a carboxy terminal end. Okay, and the side chains that come off actually come off this carbon, this central carbon here, which is actually called the C alpha carbon. And then if you want to sort of just draw it as a schematic, what you have is your polypeptide chain right here, and then you have your side chains coming off. And in this case, the book is sort of color coding them, green for nonpolar and blue for polar. And it's the sequence of these amino acids as they're added that actually helps determine the structure of the protein, and ultimately then how it comes together to form an active site to be sort of an active protein. And we'll look at that in the course of this lecture. Okay, so last time we went over the amino acids. Here they all are. You have your nonpolar amino acids, and then you have your polar amino acids, and then you have your acidic and your basic. And once again, it's I encourage you guys all to sort of memorize these. I won't necessarily test you on any specifics of them, but they're really just a good thing to know. And if you start to sort of look at things like aspartic acid is the acidic form of asparagine, right? It's very similar, just changes a little bit from the acid form to the polar form. Same thing with phenylalanine and tyrosine, very similar. Tyrosine has an OH group on it. Phenylalanine does not. A notable exception to all of them is proline, where the side chain off of the C alpha carbon actually comes around and is covalently attached to the amino terminal end. And this will oftentimes put a kink in the structure such that it can't form because it's not allowed to rotate around this bond angle here around the C alpha carbon, which leads me to the next slide, which are basically the amino acids and their bond rotation. Okay, so here you have the amino terminal end. You have your central C alpha carbon, with the side chain on it, and then you have your carboxy terminal end on this one. And so around the C alpha carbon, amino acids can actually rotate, right? They can, you can have these sort of bond rotations between the two peptide bonds. And the bonds between the alpha carbon they're just free to rotate in 360 degrees. And this basically allows the polypeptide to assume an infinite number of different conformations, right? If you think about the angles can get infinitesimally small, and you can have two of them, then there's an infinite number of conformations that a protein can take. But typically it doesn't. Typically proteins assume only one conformation. And when I say that, I want you to sort of think about the protein as a dynamic molecule, right? They can sort of move a little bit around everything, but if you were to crystallize the protein, it's basically in one conformation. And we'll see how proteins can actually undergo conformational changes throughout the course of the term, or throughout the course. Okay, so you can assume that it takes sort of one conformation, but it's actually a whole bunch of different ones, because you want to sort of think about the protein as a breathing sort of protein, okay, that can move, and they can undergo conformational changes during sort of their activity. But getting back to the Phi and Psi angles, once again, this can have a 360-degree rotation, so can this one, but unfortunately they don't. You know, the protein sort of folds into one major conformation, and if you were to map out all of the Phi and Psi angles, they could take on anyone, but they don't. And here's just the Phi and Psi angles for a single protein. Mapped around here are all of the Psi angles, and here are all the Phi angles for each one of the individual amino acids that are shown here. And one of the reasons that it doesn't assume all of the different bond rotation angles is due to the steric hindrance of the side chains. And so if we just sort of go back right here, you have this R group and this R group, and if these two bonds, the Phi and Psi around this C alpha carbon, you know, they're not going to have full 360-degree rotations, because this R group and this R group off of the protein are actually going to clash into each other, and so there's steric hindrance there that prevents them. And this occurs throughout the whole molecule and around all of the different angles, okay, extending outward even past these side chains. Another reason is that there's also intra-protein interactions, okay? And so electrostatic interactions are going to want to sort of come together to help sort of neutralize themselves. Hydrogen bonding within a protein is also very favorable. And then you're also going to have hydrophobic interactions, or van der Waals interactions, where you're basically going to get tight packing of hydrophobic molecules, or as tight as it goes. Okay, so when a protein folds, not only does the steric hindrance of the amino acid side chains come into play, but it also the electrostatic interactions between those side chains, the van der Waals interactions, attractions behind those side chains, and also the hydrogen bonding. And sort of if you want to look at it in sort of its most simplified form, if this was a polypeptide over here with polar and non-polar side chains, the protein is going to fold in such a way so that all of the hydrophobic non-polar side chains are actually packaged sort of in the middle, whereas the polar side chains would be on the outside. And this, of course, would be in an aqueous environment. Now, if it was a hydrophobic environment, the opposite might occur, where all of the sort of polar side chains are packaged in the middle, and the hydrophobic side chains are exposed to the hydrophobic environment that it would be in. So, for example, like a transmembrane protein, or a protein embedded in the membrane. Okay, so just something that's interesting about folding and unfolding, and this isn't universal, but it does happen quite a bit, where a protein just wants to assume that one conformation that it loves. Okay, and so you can take a purified protein that you can isolate from cells, and then you can denature that protein in something like urea, which makes it a string of sort of amino acids, right? A polypeptide. And then you can actually remove that urea and refold the protein to its original conformation. And you know, the vast majority of time this is going to happen correctly, but there are some proteins, especially proteins that are made in the secretory pathway, that actually have chaperones and enzymes that are needed for the protein folding and the processing of the thing. And we'll see a touch of that a little bit here. Okay, some of the major structural conformations that occur in proteins are the alpha helix. This is one of the major. The other is beta sheet, which we'll cover in just a second. And so here you can see the alpha helix, and what you have is the amino terminal end, and then here is the oxygen on the or the carbon with as it's going through, and this is the black here is the C alpha carbon with the R group coming off. And so it forms this sort of helical structure here that comes around where it's sort of looping, okay? And what you have is hydrogen bonding between them, okay? So the oxygen that comes off of the terminal carbon is actually hydrogen bonded to the nitrogen, okay? So you get this carboxyl oxygen, hydrogen bonding with the amino nitrogen, and this occurs once every 4 amino acids. So if you sort of follow this around, you can see and count them, and it occurs once every 4 amino acids. So ultimately, one complete turn of the alpha helix it occurs once every 3.6 amino acids. And this is just sort of a ribbon diagram of what the alpha helix looks like. Okay, and these are very, very common in proteins, and this is sort of just a graphical way that scientists represent this alpha helix in proteins. Another conformation that that proteins have, or folding conformation, is what's called a beta sheet. Okay, and this is where you have linear stretches of the polypeptide, and once again, like with the alpha helix, you get hydrogen bonding, and this occurs between the carboxyl oxygen and, once again, the amino nitrogen, and you can see it right here. Now, in this instance, it's sort of drawn as these sort of fat arrows. This is a beta sheet, and in this instance, it's actually an anti-parallel beta sheet where one is going in one direction and one is going in another. And this is a very sort of favorable fold because of all of the hydrogen bonding that can occur between these. Now, in the case with an anti-parallel beta sheet, all of the R groups sort of alternate between the sort of if you can sort of see my hand here, between the top and the bottom. And so here you can see these side chains are all on the top, and then these side chains down here would be all on the bottom, so they're alternating back and forth. If you have a parallel beta sheet, which we'll see in a second, they actually alternate. So 2 up, if you had parallel, it would be 2 up, 1 down, and then 1 up, 2 down, that type of thing as you're sort of going through the protein. Okay, and so here's just the example of the anti-parallel versus the parallel beta sheets, and what connects these, and actually what connects alpha helices to beta sheets and things like that, are these other domains called the loop domains. Okay, and these are sort of shaded here in green. These are sort of unstructured and can move, and if you guys remember, I said a protein will assume one structure, but it actually sort of is almost like a molten globular folded protein. It's because these loops can actually have some degree of freedom, and they can move to a small degree. Okay, so proteins also assume more of a tertiary structure. And this is when two sort of proteins can come together, or you can get instances in a single protein where, depending on how the amino acid sequence occurs in the protein, you can actually form sort of repetitive stretches. Okay, so if you have, in this instance, it's called a coiled coil, and you have an alpha helix here, and every third or fourth amino acid is a hydrophobic, whereas the other ones are sort of hydrophilic. What you'll end up with is this sort of stripe of hydrophobic domains, and they'll want to actually come together to form this coiled coil domain so that the hydrophobic amino acids are actually buried in the interior. Okay, and this sort of coiled coil is very common, and we'll see it again. The collagen, which helps to make up our extracellular matrix, actually is a 3-helix bundle. Okay, but in general, these coiled coils can occur quite a bit in different proteins. So not only are the proteins composed of alpha helices and beta sheets, but the protein can also then have subdomains. Okay, so this protein happens to be called SARC, and it has, it's actually a kinase. Here's the ATP binding domain. And then you have these other domains, which are SH2 and SH3 domains, which they got their names from SARC homology 2 and SARC homology 3 domains. All right, and here's the sort of the space-filling molecule of SARC. Okay, so there's certain level of organization in proteins. You have the primary structure, which is actually the amino acid sequence. Okay, and that you can get from the cDNA or from the DNA. You can have a secondary structure, which are alpha helices and beta sheets. And then you can have a tertiary structure, which is actually the completed fold. So this would be the tertiary structure of SARC. And then you can have the quaternary structure, where multiple different proteins come together to form a multimeric complex. Okay, and that's considered the quaternary structure. And one of the things that you need to know about this, and we'll see examples of it in the next couple slides, is that numerous proteins share the same folds. Okay, so they have these domains here like SH2 and SH3. SH2 and SH3 are not reserved just to SARC. They actually occur in other proteins, and when we get to the lectures on cell signaling later on in the term, you guys are going to actually see SH2 and SH3 domains later on. Another thing too is the kinase domain is also highly served or highly conserved. And so the question that I'm going to ask you guys, if you can think about, and name some of the different domains that that occur, and we'll see different examples of them sort of throughout, right? And we've already talked about some in the last lecture, right? Like zinc finger domains, for example. Okay, so here's just some examples of ribbon diagrams and 4 different sort of domains that are in proteins. This is a 4-helix bundle, and this comes from cytochrome B. And so you can see just the ribbon diagram of it showing the 4 alpha helices coming together, and then there's these loop domains that are connecting it. Here is NAD binding protein, lactate dehydrogenase, and this is actually composed of alpha helices and beta sheets. And one of the things I think I forgot to mention was that when the beta sheets form, due to the hydrogen bonding that's occurring, they actually sort of produce this twisting fashion where you can sort of see them sort of twisting, and that's sort of required for the hydrogen bonding to occur. And finally over here you can actually, is the immunoglobulin light chain, the variable domain of it, where you actually have 2 anti-parallel beta sheets sort of coming together to form sort of like almost like a sandwich. Okay, and when it binds, when immunoglobulins bind antigens, and so what it does is it tends to bind the antigen in this region up here with the loop domains are. Okay, and the anti-parallel beta sheets are sort of what holds the loop domains there. And once again, here you also want to sort of note the twist that's occurring in the beta sheets. Okay, so one of the things you have to realize, we have a pretty complex genome, right? There's estimated we have about 20 to 25,000 different genes, and these sort of arose through evolution and gene duplication, a lot of them. And then all of a sudden an enzyme can take on a new function. So here's elastase and chymotrypsin. These are actually both produced by the pancreas and are digestive enzymes. And they have a very similar active site, and we'll see this in a couple of slides. And the protein domains are very similar, but they actually have completely different functions. And so even though they're very similar, they sort of evolved through gene duplication, where chymotrypsin or elastase was actually duplicated, and then through sort of evolution, new amino acids were changed with different amino acids, and you get a protein that's folded very similar, and the active sites are very similar, but the proteins actually have different functions. And so shared domains occur quite a bit. In this section, this is actually called the homeodomain right here, where you have one helix, a loop, a second helix, and a third helix. Here's the underlying amino acid sequence of two homeodomain containing proteins, one from yeast and one from Drosophila. And you can see that there's a lot of amino acid divergence in these, yet the protein fold is almost a complete overlay. Okay, helix 1 here is green, helix 2, and then helix 3. And just a little note, this protein called engrailed is also a homeodomain protein, and I bring this out because this is actually one protein that I know of that has been implicated in autism spectrum disorders. It's a transcription factor, and so I just wanted to highlight that for you guys. And so it's estimated that there's about 2,000 different folds, and 800 have thus far been cataloged. And actually 50 of the most popular domains actually occur in 3/4 of all the proteins that are out there. And when it comes to structural biology, and before I get to the next slide, I just wanted to mention Rutgers University houses the Protein Data Bank. This is where all of the protein structural biology data is stored in a big database. And if you guys are ever over on the Bush campus, you walk into the proteomics building over there, you can see it. And the PDB is actually a great resource. You can log on, pull up your favorite protein, see if the crystal structure has been solved, and then sort of play around with it. And you can sort of view it in this sort of structure and see if you can sort of gain some information about its function. Okay. Another way that people use to sort of study proteins is using this program called the BLAST search. Okay. Now, in the sort of the post-genomics age, there's actually hundreds of thousands of sequenced genomes from all different organisms, and these are all stored in databases. NCBI is one particular database where you can get sort of a whole bunch of different sequencing data. And NCBI also has this program called BLAST, where you can take in a protein that you may be working on, and you can blast it against a whole bunch of others and find sequence similarities, okay? And oftentimes you can then sort of figure out whether or not there's sort of structural similarities between the two. And these can help you sort of predict the domain structures and possibly protein function. Okay, so the point here is that there's a lot of bioinformatics packages out there for you guys if you're ever studying a specific protein. Once again, sort of protein domains are shared, so you can have a single domain, but yet many proteins, many different proteins have that same domain. So in this case, here's EGF, epidermal growth factor, and this one domain of EGF actually occurs in urokinase and in factor 9 here. You can have also chymotrypsin has this domain, and it also occurs in urokinase, plasminogen, and so you can just sort of see here the different protein domains are shared with different proteins, right? And this actually has to do with gene duplication and recombination, and then some domain shuffling that can occur. And we'll talk about genetic recombination in some upcoming lectures. Here are just some common domain modules. You have the complement control module here, fibronectin module 2. Here's a growth factor module. Here's a immunoglobulin module, fibronectin, which actually looks very similar to the immunoglobulin module. Then you have a Kringle module over here. So there's all different kinds of protein domains. They could have put the SH2 and SH3 domains here as well, which we saw in SARC. There's also domains called like PhD domain, plextrin homology domain, EP1 and EP2. And so what you end up getting is sort of these inline duplications that can basically lead to a repeat of the domain, or certain recombinations can put a gene next to another domain from another protein, and that can give rise to a whole new protein function, right? And as the amino acids change, the enzyme can evolve. And so here's an example of fibronectin where you get this same domain that's actually repeated 4 times. Okay, and this is just an extracellular protein with the fibronectin domain repeated. And here's just some examples of a different protein in yeast, worms, and humans, and how the domains can actually be sort of expanded by duplication and recombination into different proteins. And here you have one that actually has gained a zinc finger domain. Okay. Okay, when we get back into quaternary structures, you can see that many enzymes are composed of multiple polypeptides, and these are arranged tightly by non-covalent interactions. Okay, and in this example, it's just a dimer. It's the CRO repressor from bacteria, and you can see that it's just a dimer that's identical, and it's sort of attached together through non-covalent interactions. Okay, and in this case, it's a shared beta sheet between the 2 proteins. Okay, and these, for what it's worth, homodimers in proteins will always have at least one axis of symmetry. Usually there's multiple axes of symmetry, including rotational, but you can find these these symmetric axes in a number of proteins. And that happens with tetramers as well. So here you have hemoglobin, here's the central heme and the 4 different subunits, and they all come together, and they're held, these different subunits of hemoglobin are all held together with non-covalent interactions. Same thing happens with the tetramer neuraminidase. And once again, as with heterodimers, heterotetramers or homotetramers have and heterotetramers and homotetramers actually have multiple axes of symmetry. Okay, and you can see the rotational symmetry in neuraminidase here if you just sort of follow this blue circle around. There's also a wide variety of sizes of proteins and large, multi-subunit proteins. And if you guys want to think about the ribosome as being, an RNA protein, large RNA protein structure, if you can compare just that to sort of like an SH2 domain, it's huge. Okay, here's catalase, lysozyme. Here's collagen, which we'll see in a second. That forms a 3-helix bundle. Deoxyribonuclease bound to DNA, cytochrome C. Here's a porin molecule, alcohol dehydrogenase, chymotrypsin, calmodulin. Here's the small insulin, and then you have aspartate transcarboxylase. So the big take-home message here, and I don't expect you guys to memorize all of these, but it's good to really sort of just know that proteins, different proteins have different sizes and different shapes depending on, what they're used for. You can also have repetitive structures in proteins. It's where you have just a single free subunit, and then it can assemble into, in this case, it's a homodimer, or you can get repetitive arrays where you can sort of form a helical, or you can have sort of like a ring shape. Okay, and there's many large structures that are sort of composed of these repeating subunits. When we look at the cellular cytoskeleton, we'll see that both actin and tubulin sort of buy into this whole thing, where tubulin forms a helical tube and actin forms these little filaments. And here's actually the actin filament right here. It can get quite large, and when it's assembled, it too has directionality. Okay, so here is a scanning EM of 2 actin filaments here, and you can have a sort of a plus end and a minus end in these. And we saw this in the Harvard movie with some of the actin filaments. And then there's actually motor proteins that we'll learn about that can actually move on these filaments to help give the cell some of its dynamic properties. Here's the collagen fiber. This is a protein fiber that actually is very prevalent in our extracellular matrix. Here's just basically a short little cross section of it, and then when you sort of zoom down in, it forms this collagen triple helix bundle here, which is very similar to the 2 helix bundle that we saw, except there's 3 of them. Okay, and just in general, fibrous proteins are quite common in the extracellular matrix, right? It's what sort of gives us our rigidity. But we also stretch, right? And so if you guys have ever wondered what allows your skin to stretch, it's actually this protein molecule called elastin. Okay, and so here it is, you've got this elastic fiber which can actually be pulled, but it's sort of all held together and doesn't pull apart because it has these crosslinks. Okay, so it can be disordered in one, and then it can be sort of pulled, and it forms this sort of cross-linked mesh like protein matrix. And the cross-links are actually this a specific covalent bond called a disulfide bond, and this happens with free cysteines in the polypeptide chain. So if you guys remember, the amino acid cysteine has an SH group, and when you're in the cytosol, which is actually a reducing environment, the cysteines are just sort of free SH groups. Okay. However, when you enter the endoplasmic reticulum, which is part of the secretory pathway, and that's an oxidizing environment, what ends up happening is you get these disulfides forming, where these proteins can come together, and you can have an intrachain or an interchain disulfide bond. So this is actually connecting 2 different proteins together, whereas this is connecting just between the one. And so in the case with elastin, these disulfides are actually the interchain. They occur between 2 different elastin molecules, and that helps connect them together. The enzyme that forms these disulfides in the endoplasmic reticulum is a protein called PDI, and we'll see PDI come up again, and I'll remind you guys of it when we get to the secretory pathway, which is in the sort of the third half of the course. And one of the things with these disulfide bonds is they increase the rigidity and sort of help to stabilize protein structures. Okay, so getting back to repetitive arrays, you can have a single subunit that actually forms a repetitive array, and you can get sort of like a hexagonally packed sheet or a helical tube. Once again, tubulin is going to sort of form this helical tube. And these things can then go on to form sort of like supramolecular structures, right? The extracellular matrix, tubulin, and viral coats. And so here's some example of some scanning EMs of different viruses. and you can see the proteins all sort of come together in these repetitive arrays where they just keep forming the same repetitive array over and over again. Okay, and here's how some viral coats actually assemble, where you have your your capsid protein, and this can have an RNA binding domain associated with it. So this protein's made, then it can sort of self-associate to form a dimer, and then the free dimers start to come together, and then you start to get the formation of an incomplete particle. If it's an RNA virus, as this one is, because it's got an RNA binding domain, it will package the RNA, and then you eventually get the formation of this repetitive array that's an intact virus. Okay, and it all comes off of this single capsid protein, comes together to form this repetitive array. Here's an example of that. It happens to be the tobacco mosaic virus. This virus happens to infect the tobacco plant, and you can see it forms this cylindrical tube, much like what was shown here, right, where you get the helical tube. And here's just a sort of a space-filling model of the EM that's here. When I say EM, I mean electron micrograph, for those of you guys who don't know. Okay, so I said before that many proteins will self-assemble, although there are exceptions, and certain proteins, once they're sort of translated on the ribosome, they get processed. Okay, and an example that your book uses is insulin, but this happens with many other proteins, and we'll see this throughout the course of the term in certain examples. Okay, so here's basically the amino acid sequence of proinsulin. It actually gets co-translationally translocated into the endoplasmic reticulum, and once it's sort of in the endoplasmic reticulum, the enzyme PDI, or the PDI homolog, forms these sort of disulfide bonds, and then it actually gets cleaved. Okay, the peptide gets cleaved to become sort of become basically a complete insulin molecule. And then when you want to get rid of the insulin, you can actually reduce the disulfides that are formed in the protein, so you're basically left with these two individual peptides. Okay, so the peptidase that's sort of cleaving this from here to here is what makes proinsulin, what helps make proinsulin into its active insulin form. Okay, and so you can then sort of reduce these and they get it going. So ultimately there's a lot going on when a protein is co-translationally translocated into the endoplasmic reticulum. You can get the protein folded, processed, and it can get glycosylated and then transferred out through the secretory pathway. Okay, so proteins often come together with their ligands, okay, and protein-ligand binding is actually a really important concept. Okay, so here you have your protein, it has a binding site, then you have your ligand, and what happens is proteins have sort of selected through evolution their specific ligand by the availability of various non-covalent interactions. Okay, and they also sort of create sort of a conformation where the ligand can actually fit. You can almost sort of think of this as a hand in a glove, right? A hand can go into a glove. And not only is it the non-covalent interactions, but the ability of the ligand to fit within the binding pocket. These constraints help to sort of maximize ligand specificity, okay, and basically tell what protein is going to fit, what ligand is going to fit into what protein. Proteins also fold so that their active sites are available to sort of create a sort of a catalytic center. Okay, so here you have your primary amino acid sequence with all of the different side chains on the amino acids, and when it's ultimately folded, the amino acids that actually make up the active site or the binding site will be sort of co-localized together, and these will allow all of the non-covalent interactions or the catalytic activity to occur within the protein. And so here's just an example of a cyclic AMP binding protein. Cyclic AMP is shown here in green, and here are all the different non-covalent interactions that are occurring with cyclic AMP along the sort of primary amino acid sequence in the fold of the protein. Okay, so these are all the different amino acid side chains that are here, and the hydrogen bonding that's occurring, and the electrostatic interactions. All right, so this is a real-world example showing a cyclic AMP binding site. And so just take a note of the hydrogen bonding and the electrostatic interactions that can occur. Now, oftentimes the binding site can also influence the chemistry, okay? And so you can have amino acids that are a little ways away from where the active site would be, but these can actually influence the chemistry. Okay, so in this case what we're looking at is the active site of chymotrypsin and elastase, and they have these three shared amino acids, the aspartic acid, histidine, and serine, and the serine is the reactive amino acid, but it's the aspartic acid which helps to draw a proton off of the histidine, and then the histidine draws a proton off of serine to make the serine active. Okay, so you can get these hydrogen bond rearrangements within a protein to sort of make it catalytically active. Okay, so amino acids that are actually away from the active site can contribute to the enzymatic activity of the protein. Now, active sites can actually differ even if the tertiary structure of the protein is conserved, right? So you can have domains, and we saw this sort of with elastase and chymotrypsin, where they had very similar folds, but the active site and the active site was different. The active site was largely the same, but they had different activities. In this case, what you have is the same tertiary structure, but the active site can be a little bit different, okay? And in this case, it's a phosphotyrosine, and here you have a polypeptide ligand. And so phosphotyrosines, can come into play, and they're very important in cell signaling, which we'll see later. Okay, so the active sites of enzymes with similar function can have a large degree of conservation, I guess is the take-home message of this slide. So I'll leave it at that for now for part one. Part two, we'll just continue on with what we're doing here with proteins. Okay.